again and welcome to ASL level biology classes. Today this is going to be the lecture 4 from chapter 1 cell structure. In the previous class, if you remember, we have discussed about the cell structure, various cell organelle, that is nucleus and endoplasmic reticulum. In today's class, we will be learning about the cell organelle, that is Golgi apparatus in detail, and lysosomes. Let's get started with the Golgi apparatus. Now, this is how the Golgi apparatus looks like. The series of flattened sacs that modifies, packages, stores, and transport materials out of the cell. The stack is constantly being formed at one end from vesicle, which bud off from the ER and broken down again at the other end to form a Golgi vesicles. So from here, if you see, from here the vesicles form from one end, and to the other end, the vesicles which are coming from the endoplasmic reticulum, rough endoplasmic reticulum, it joins to the Golgi apparatus. So the main function of the Golgi apparatus is that it modifies, packages, stores, and transport materials out of the cell. Two examples of protein processing in the Golgi bodies are the addition of sugar. So the two processing which is happening in the protein processing which is happening inside the Golgi apparatus is addition of sugars to make proteins molecules known as glycoproteins. So whenever the sugar is being added in the protein, we call them glycoprotein. Glyco means the sugar and proteins. And the removal of the first amino acid methionine from the newly formed protein to make it a functioning protein. So the second thing is that we have to remove the Golgi apparatus, remove the methionine amino acid from the protein. So removal of amino acid protein from, that is methionine, from the newly formed protein. So these are the two kind of processing that happens in the Golgi apparatus. So the protein which is formed in the endoplasmic reticulum, that it comes out from the rough endoplasmic reticulum in the form of vesicles. It goes and joins with the Golgi apparatus, and inside the Golgi apparatus, the modification happens. That is, there can be addition of sugars, there can be like formation of glycoproteins, or addition of lipids to form a lipoproteins. There can be removal of the amino acid, the first amino acid, that is methionine, which we will be talking when we will see the protein synthesis. The first amino acid which is added in the protein in the translation process is the methionine, and that has to be removed. So in order to make the protein functioning, so that is again done in the Golgi apparatus. And in plant, enzymes in the Golgi body convert sugar into cell wall components. Golgi vesicles are used to make lysosomes also in animals. So in animals, the Golgi vesicles form lysosomes. The next cell organelle that we will be talking is lysosomes. Now lysosomes are spherical sacs surrounded by a single membrane. So it's not a double membrane, it's a single membrane having no internal structures. They are commonly around 0.1 to 0.5 micrometer in diameter. That is the diameter of average diameter of a lysosome. And they contain digestive hydrolytic enzymes. Digestive enzymes like hydrolytic enzymes. Lysosomes are responsible for breakdown, that is digestion of unwanted structures such as old organelles and even the whole cell. So very important, these lysosomes are very important when it comes to removal of something which is not required in the body. So lysosomes, they just simply digest that cell. If the entire worn out cell can be digested by lysosomes, and even old organelles, and certain bacteria also, microorganisms also. The head of the sperm contain a special lysosome called as acrosome for digesting a path to the egg. that is ovum. So during the fertilization, when the sperm enters into the egg, it has to digest the membrane present in the egg in order so that it can the sperm, the genetic material can go inside and fertilization can happen, the nucleus can go inside. So that can only happen when the membrane gets digested, and it gets digested by a, a, a enzyme, hydrolytic enzyme, which is present in acrosome. So it's a special kind of lysosome only. So this is how the lysosome looks like. It's a spherical structure, as you can see, having a single membrane, and inside what you see is the hydrolytic enzymes. These are hydrolytic enzymes, what you see here. And the pH of these hydrolytic enzymes, the pH of these lysosomes are generally low pH. This basically helps these hydrolytic enzymes to be activated. And apart from this, there will be transport proteins on top of these membranes, which help these lysosomes to move. With this, we have finished two cell organelles, and I hope you have understood the two organelles that we have discussed, that is Golgi apparatus and lysosomes. Thank you, and in the next class we will discuss other cell organelles in the animal cell. Thank you.